For this topic, we are going to discuss the visual system. So we're going to start with the physical energy for the visual system. Okay. Just remember that no species can see in the dark, but some are capable of seeing when there's very little light. Okay. Light can be thought of as particles of energy, which we call photons, or waves of electromagnetic radiation. And you'll notice in the graphic the different wavelengths here. So visible light is only a certain part of the spectrum that you notice here. Humans see light between 380 and 760 nanometers. So that is visible light. We cannot see gamma rays. We cannot see radar or infrared rays. Okay. Wavelength is the perception of color, and intensity is the perception of brightness. Okay, let's start with the sensory organ. Now that we know what the physical energy is for vision, now how does it get into our body so that we can identify and perceive things that we see? First, the light has to penetrate through the cornea, which is a protective outer layer that aids in controlling and focusing light. That light feeds through the pupil, which is the hole that allows light to strike the retina. It also changes in size based upon illumination. Then it passes through the lens, which focuses the light on the retina. And what moves that lens back and forth are the ciliary muscles, okay, which alter the shape of the lens so that we can see things up close and see things far away as well. And then accommodation is the process of adjusting the lens to bring images into focus. Okay. So now what we're going to do is looking at the back portion of the eye, or the retina. Okay. The retina is actually built in a sense inside out, so that light passes through several cell layers before reaching its receptors. The photoreceptors, as you see in the picture, they're the ones at the bottom there, are the rods and cones. Okay, and these sensory neurons detect light. They are the transducers of the visual system. They take that physical energy and turn it into something that can be understood by the brain, an electrical chemical signal that's understood by the brain. We also have bipolar cells, which receive the input from the photoreceptors, and synapse on ganglion cells, whose axons form the optic nerve. Okay, and so that's what leaves the eyeball itself, is that optic nerve on its way up to the brain. There's also lateral communication that happens in the retina. Okay, the horizontal cells, as you see also in the diagram, aid in communication between photoreceptors and bipolar cells. And amacrine cells aid in communication between bipolar and ganglion cells. So the idea here is that there are a lot of cells involved, okay, because our visual sense is just amazing. It's spectacular. What we're able to perceive in the real world is pretty amazing. All the different colors that we see, the details that we see, so it takes a lot of communication, and you see this in a vertical sense and also in a lateral sense. Now, this is another view of the retina, okay? But I wanted to focus on a couple other terms here. Blind spot, okay? So no receptors exist where the information exits the eye, okay? The ganglion cell axons, that optic nerve, when it leaves the eyeball, there's no receptors there, so that's a blind spot. Well, why don't we see the blind spot? Well, our visual system uses information from cells around the blind spot for completion, okay? And also we have very quick eye movements called saccades. And these are, our eyes are constantly just moving a little tiny bit so that we aren't focused straight ahead and notice that blind spot, okay? Also, I wanted to bring up the fovea. The fovea is the high acuity area at the center of the retina. So if you want to place your best sight on an object, you're going to put it right in front of your eyes, because that's going to pick up on the fovea. You'll also notice in that picture that there's a thinning of the ganglion cell layer, which reduces the distortion due to cells between the pupil and the retina. Okay? It's just cones. And that's another part of this also, is that the fovea only consists of cones, okay? Very high acuity and for color. Let's talk a little bit more about just the rods and cones, okay? The duplexity theory of vision. The duplexity theory means that there's two of these different types of cells. There's cones and rods. Cones are what we refer to as the photopic system. They're used for daytime vision, very high acuity and color, okay? Rods, or what we call the scotopic system, which is nighttime vision, high sensitivity, allowing for low acuity vision in dim light, okay? So high sensitivity to any light that's out there. They're very good in dim light, but they don't have that color aspect to them, okay? There's much more convergence in the rod system, and you can see that in the graph that I provide you there. It says high convergence in rod-fed circuits. You see how there's one ganglion cell, okay? Or I should say, start from the right-hand side. There's 1, 2, 3, 4, 5, 6, 7, 8. There's 8 rods there, but you see how each one of those rods are converging onto a bipolar cell and then onto a retinal ganglion cell, okay? Low convergence is seen in cone-fed circuits. So now up at the top, you see a cone, which is only influencing a bipolar cell, and that bipolar cell is only influencing a retinal ganglion cell. So less convergence there, okay? But it aids in the high acuity sense of a cone, okay? And then that last point there, again, only cones are found at the fovea, like we mentioned. Okay, more detail for you. Approximately how many of these cones do we have? Well, in the table there, it says 4 million. Okay, but for the scotopic system, rods, we actually have 100 million, many, many more. Okay, the photopigments, 3 classes of cone opsins, the basis of color vision, and we'll talk about that in a little bit. For the rods, only rhodopsin is the photopigment. For sensitivity, cones, low, needs relatively strong stimulation used for day vision, which means that you need that light for that high acuity vision. Okay? For rods, high sensitivity can be stimulated by very weak light. Okay, so good for night vision. The cones concentrated in and near the fovea. For the rods, outside the fovea. For cones, the receptive field size, small in the fovea, so acuity is very high there, larger on the outside of the fovea. And then for rods, larger, so acuity is lower. Okay? And then finally, the response time. For cones, it's relatively rapid, but for rods, it can be slow. And we have all experienced this from going from a very bright environment into a dark environment, and you can't see. And it takes a little while for you to, for those rods to react to the low light. Okay. Now, if we talk about these rods and cones from a chemical point of view, we see that, okay, on the left-hand side is a rod, on the right-hand side is a cone. So let's focus on the rod first on the left. It says rhodopsin is a pigment and a G protein linked receptor that responds to light rather than neurotransmitters. Okay, so in the dark, sodium channels remain partially open, releasing glutamate. And you'll see that on the left-hand side, it says in the dark, and then at the very bottom, rods continuously release glutamate. Remember, glutamate is an excitatory neurotransmitter, so it's going to cause things to happen. And it also has that word in there, depolarization again. Depolarization means that there's a likelihood for another action potential, right? So action is taking place here. But when light strikes that rod, the sodium channels close, okay? So sodium's not coming in, then the rods hyperpolarize, and then they inhibit the glutamate release. So you look at the bottom in the light on the right side at the bottom, number 4, it says glutamate release is reduced, okay? So that's the basis behind the rods. For the cones on the right-hand side, you'll notice also we're dealing with glutamate. Okay, that glutamate is released. from the cone if there's illumination, okay? If there's light around, then they're going to be activated. And they could activate an on-center bipolar cell, okay? And if it's on-center, which means it's focusing on, it kind of depends on the shape, the size, the color of an object. But if it's one that activates it, then it depolarizes it, and you see all that glutamate released into the ganglion cell, and therefore increasing the firing rate. Okay? On the right-hand side, if it's an off-center bipolar cell, okay, then it's going to cause it to hyperpolarize. There'll be less glutamate released to the off-center ganglion cell, and so a decreased firing rate. And we'll talk a little bit more about those on-center, off-center, and complex bipolar cells in just a minute. Okay, the eyes work over a wide range of light intensities. If you look at the graph here, at the bottom, it's rod vision, okay? Rod vision because rods are good in dim light, right? So that's where they're activated. Then as you add light to your environment, then the cones take over, okay? Because those are the ones that are specific for color and specific for high acuity. So how does this work? Well, range fractionation uses different photoreceptors to handle different intensities. Okay? So that's how you see once those cones begin to activate, you'll see different photoreceptors that handle the different intensities of light. And then in photoreceptor adaptation, each photoreceptor adjusts its level of sensitivity to match the average ambient level of light. So, and we're talking about photoreceptors here, so rods and cones. So low light, the rods are going to be activated. A lot of light, substantial light, then the cones are going to be activated. Okay. All right. Now what we're going to talk about is from the retina, we've focused all of it on physical energy coming into the eyeball. We know that the rods and cones are now the transducers for this physical energy, but how does it make its way up to the cortex? And here is the pathway, the retinal geniculate striate pathway. Okay. So first, ganglion cell axons from the retina form the optic nerve. The optic nerve crosses over at the optic chiasm. These axons are now in the CNS, so it's called an optic tract, not an optic nerve anymore. And we learned the difference between those terms. Nerves are what we call it in the peripheral nervous system, but in the central nervous system, we call these optic tracts. The axons from the optic tract then terminate on cells in the lateral geniculate nucleus, which is in the thalamus. Remember, the thalamus is the switching station. So it's saying, wait a minute here, this is visual information. I need to send this visual information to a particular area of the cortex. And what is that particular area? Well, that is the primary visual cortex in the occipital lobe. Okay. The primary visual cortex is also called the striate cortex. Okay. And then some visual information is also sent to visual association cortex, which is also known as the extrastriate. So that is the pathway for vision from physical energy through the eye, through the retina, to the optic nerves, the optic tract, or optic chiasm, optic tracts, to the thalamus, to the primary visual cortex. Okay. Information received at adjacent portions of the retina remains adjacent in the striate cortex. This is something that we call retinotopic. Okay. What this means is that it's a mapping. The retina image or the physical activity, the activity in the retina is mapped to the primary visual cortex in a very similar manner. Okay, so it's retinotopic. The retina is like a topographical map in the primary visual cortex. So more cortex is devoted to areas of high acuity. That makes sense. Okay. About 25% of primary visual cortex is dedicated to input from the fovea, because the fovea was the highest acuity area of the retina. Remember, that was the area that only had cones involved. That was the area where if you really want to focus on an object, you put it straight in front of your eyes, okay? Because that information is going to be fed back to the fovea. Also, retinotopic mapping makes it possible to predict the location of a scotoma or a perceptual gap due to an injury. So if there's an injury to the retina and the individual has a blind spot, okay, which is what this scotoma looks like, then that's going to be mapped back to the primary visual cortex as well. Okay. And then blind sight describes the inability to consciously perceive visual cues, yet be able to tell where the object is. So they know where the object is, but they can't name that specific object. So I just wanted to put in a couple of these issues that come up with the visual system. Okay, now that we're focused on the cortical area, or mostly the cortical, that's where we're going now. We're going to pause for a moment and going over seeing edges. Okay, it's very important in our environment that we see edges. That's how we know that one thing ends and the other one begins. That's how we know that one object is in front of the other object. Okay, so you can see the graphic here, and they call those mock bands. It looks as though there's a line between each variation of blue, okay? When in actuality, there is no line there. That's what our visual system is building in, okay? From the retinal level, on the left-hand side, it's called lateral inhibition. Sensory receptor cells inhibit information from neighboring receptor cells, producing a contrast effect at the edges of regions, okay? So, and that's at the retinal level. At the cortical level, on the right-hand side, you have simple cortical cells called bar detectors or edge detectors, and they respond to an edge or bar of a particular width, orientation, and location in the visual field. Also, you have complex cortical cells, which respond best to a bar of a particular width and orientation that is in motion anywhere in the visual field. So let's look at these a little bit more in detail. Okay, here's some cortical cells on top. A simple cortical cell is sensitive to orientation. You notice here that the line that is vertical, and then you look to the right, you see all the different little hash marks, okay? That's the firing of that simple cortical cell. So you'll notice that the firing is very rapid for a vertical line, but not very rapid at all for a horizontal line. At the bottom, you have a complex cortical cell, and it's also sensitive to motion. So not only direction, but motion. This particular cell responds strongly only when the stimulus moves down. So it's going to be the one at the top. So it has to be a horizontal line, and it has to be moving down, and then you'll see how rapid that firing rate is. So there's millions and millions of these cells, these cortical cells, that are picking up all the different attributes of what we see. The primary visual cortex doesn't deal in images, it deals in different small components, vertical and horizontal lines, and so on. Okay, so up to this point, we've only mentioned that the information for the visual system leaves the lateral geniculate nucleus of the thalamus and goes to the primary visual cortex, also known as striate cortex. But that information can also go to pre-striate, okay? It can also go to the posterior parietal cortex and also to the inferior temporal cortex. Okay? And so on the left-hand side here also, I provide you a graphic that shows all of these different areas, okay? V2, V4, and the inferior temporal lobe are also shown on there. And we could go into a lot more detail on exactly what V2 and V4 does, but it's outside of the topic of this class. Okay, a couple other things at the cortical level here is there's different pathways, okay? And we think that these are listed, well, they're listed as where versus what theories, okay? There's a dorsal stream and a ventral stream. So let's start with the dorsal stream. says pathway from the primary visual cortex, and you'll notice that as the red arrow going upwards. Okay, pathway from primary visual cortex to dorsal pre-striate cortex to the posterior parietal cortex. You see, that's exactly where it is. Posterior to that central sulcus there, where you see the somatosensory cortex just behind that. The where pathway, the dorsal stream, okay, location and movement, or pathway for the control of behavior, reaching for an object. So the dorsal stream, when this information leaves the primary visual cortex, it goes up to the parietal cortex. We think that's the where pathway. Okay? The ventral stream is where the information leaves the primary visual cortex to the ventral, right? That's at the bottom, ventral pre-striate cortex, to the inferotemporal cortex. So inferior is going to be at the bottom of the temporal lobe, and that's the green line that you see there. And we believe that this is the what pathway, responsible for color and shape, or the pathway for the conscious perception of objects. And I put in here a term for you. This is prosopagnosia. This is the inability to recognize familiar faces. It's a pretty fascinating phenomenon here. These people don't have issues with their vision. They can see the vision, but they don't recognize faces. It's very, very specific. Okay? And we believe it's associated with the damage to the ventral stream between the occipital and temporal lobes. So that gives more basis to that ventral stream and the what pathway. Okay, let's talk about color vision. So color is created by the visual system as we detect differences in the wave-like movement of photons through space. And we already talked about those in nanometers and the range for visible light. Okay, differences in wavelength allow us to perceive different colors within a certain range. An object's color depends on which wavelengths are reflected and thus perceived. The other colors are absorbed. Okay, so 3 dimensions of color perception. First, brightness varies from dark to light, and we discussed that. Hue refers to the origin of the colors we can see. So primary and secondary colors, for instance, yellow, orange, red, violet, blue, and green, are considered hues. However, tertiary colors, which are mixed colors, where neither color is dominant, would also be considered a hue. So we haven't talked about hue and saturation yet, okay? Only in the aspect of color vision do we bring these up. And then saturation varies from full color to gray. Right? So some hypotheses here for color vision. Color perception involves receptor cells with different spectral sensitivities. One hypothesis is the trichromatic hypothesis. This hypothesis believes that 3 different types of cones exist, blue, green, and red. Each responds to a specific different part of the visual spectrum. Each has a separate pathway to the brain, and the color is recognized based on which receptors are activated. So very, very straightforward. There are cones that are blue cones, and they send that pathway to the brain, and it's recognized by the cortical area as blue. Okay. There's also the opponent process hypothesis, which views 4 unique hues, blue, green, yellow, red. Okay. And then 3 opposed pairs of colors. So there's the opposing colors are blue, yellow, green, red, black, white. So 3 physiological processes with opposed positive and negative values are the basis of color vision. So which hypothesis do we go with? Well, both. Okay. Both hypotheses are part of current color vision theory. So first, each cone contains one of 3 pigments, and we saw those, okay, called opsins, which have overlapping but different peaks of sensitivity. So on the left-hand side, it says cones are named for peak area of wavelength. Short, you see that blue, okay, medium, it's going to be in green, and then long is going to be I guess, in a kind of a yellow area. Okay. And then most objects stimulate at least two types of cones because we see all different types of hues and saturations of color. We very rarely see just a pure color. The nervous system compares the degree of activation across receptor types and extracts color information. And then certain ganglion cells and higher level cells are color specific. So they're going to feed that color back to the brain.